Um, so let's start. Welcome everyone. This is the last Tech Talk for 2023. The Tech Talks are brought to you by the Monash Partners Comprehensive Cancer Consortium, the MPCCC. My name is Cho, and I'll be your host for today's Tech Talk. So to begin, I would like to pay our respects to the traditional custodians of the lands from which we join together today, and acknowledge their elders past, present and emerging, as well as any members of the Aboriginal and Torres Strait Islander community who may be among us. So to set the scene, the Tech Talks aim to provide an informal and interactive forum for our professional research community to cover and discuss the latest cancer research technologies and how they can be applied in a practical way to support our research. The Tech Talk format offers plenty of time for Q&A, and we really encourage you to tell us about your experiences, key learnings, tips and tricks, as well as any potential challenges and limitations. So today I'm very, very pleased to welcome Dr. Claire Sun and Dr. Paul Daniel. They'll be discussing the Childhood Cancer Model Atlas. So Claire is an early career researcher and an emerging research leader in computational biology and cancer research. She's the current recipient of a Victorian Cancer Agency Early Career Fellowship, and since 2018, she's been the lead bioinformatician in the Next Generation Precision Medicine program at the Hudson Institute of Medical Research. And Paul is the senior postdoc in the Next Generation Precision Medicine program. They're the co-first author on a paper in Cancer Cell that reported on the Childhood Cancer Model Atlas, or CCMA, and they will present this to us now. So for the presentation, could we please ask you to please mute your microphones and turn off your video to minimize disruptions, and we'll take questions after. So thank you very much, Claire and Paul. I hand over to you now.
Thanks for the introduction. As mentioned, Claire and I are both postdocs in the Next Generation Precision Medicine program. So we're located at the Hudson Institute of Medical Research near the hospital down the road from the Clayton campus. So our team's actually around 25 strong and led by Professor Ron Firestein. So today we'll be talking about our efforts over the past 7 years, where we've established a living biobank of childhood cancer models, which we believe will serve as a key resource for efforts going forwards. Oops. So our lab works primarily on high-grade solid pediatric cancers, which is a type of malignancy which develops in children from 0 to 14 years of age. Although it's a relatively rare occurrence, childhood cancer is the #1 leading cause of death by disease in children, much greater than things like infectious disease or congenital diseases. It's estimated by the World Health Organization that over 400,000 children worldwide are diagnosed with cancer every year. And in Australia, around 1,000 are diagnosed every year, with about 3 children dying every single week from childhood cancer. The most common type of childhood cancer is leukemia, followed by brain cancer and then lymphoma. For leukemia, there's actually good news in that there's been quite rapid improvements in survival over the past few decades. For instance, in ALL, it now represents one of the most curable cancers, with the 5-year survival exceeding 90%. And as you can see, there's been quite a steady increase in survival over the past few decades. And it's really the genomic era of medicine which has been largely responsible for this, as we're now able to identify precision medicine opportunities in these patients to give the right drugs to the right patients. Unfortunately, that's not really the case for a lot of other cancers, childhood cancers, especially childhood CNS cancers. So for example, if you look on the graph to the left, if we compare 5-year survival rates for some of the most common pediatric cancers, we can see diffuse intrinsic pontine glioma down here at the bottom with a 5-year survival of under 1%. And this has really been because there hasn't really been advancements in the way we treat these cancers over the past couple of decades. So if you look at the graph on the right, we can see compared to leukemias, where over the past couple of decades, there's been about a 20% increase in 5-year survival. For CNS cancers, there hasn't really been an equivalent increase in survival. And due to this, despite brain cancer being diagnosed around 3 times less in pediatric populations, brain cancers have recently overtaken leukemia as the leading cause of death in children under the age of 14. So this lack of improvement for many CNS cancers is because, as childhood CNS cancers, is really because for a long time they were just considered to be very similar to their adult counterparts. And indeed, if you compare the pathology and disease progression of many pediatric disease, they do look very similar. However, we now know in the genomic era that they're quite molecularly distinct. And we now know that they really require quite a unique approach for effective treatment. Despite this, the way that we approach treatment for many CNS cancers have remained the same since about 1960. And we still use quite cytotoxic therapies such as chemotherapy and radiation, despite the fact that we know that patients which do survive these therapies have long-lasting effects. For example, patients which receive radiation or chemotherapy have a 5 times greater increase in chance of developing secondary cancers compared to control populations. So these really emphasize the need for greater efforts in research to identify effective treatment options for these pediatric cancers. So towards this, cell line models really are foundational resources which researchers use to enable investigation of these sorts of things, because we can use them to identify aspects of tumor development as well as mechanisms of disease progression to things such as drug discovery and biomarker identification. However, when we look at the availability of cell lines in the literature, there's a much greater representation of adult cancers compared to pediatric cancers. So there's thousands of adult cancer cell lines available, representing all the different types of varied adult cancers. Whereas when we compare it to the availability of pediatric cancer, there's only a couple of hundred in comparison, and there's quite low representation of some of the rarer types of pediatric cancers. So this is really where our program started, and in an effort to sort of address this discrepancy, the Next Generation Precision Medicine program started about 7 years ago to generate a single-site collection of childhood cancer cell line models, which undergo comprehensive molecular and functional characterization, which we have released and called the Childhood Cancer Model Atlas. So the schematic at the bottom really shows how we've gone about generating the CCMA. Essentially, when a child is diagnosed with cancer and goes into surgery, part of the tumor is resected and given over to either a local lab or sent to us, where we establish viable cell lines. After establishing a viable cell line, the cell line is sent for a host of different molecular characterizations, including whole genome sequencing, methylation and transcriptome sequencing, as well as functional characterization using high throughput drug screens and CRISPR-Cas9 knockout screens. And together these enable the generation of a multi-dimensional profile of each cell line, which we hope serves as a foundational resource with which to advance childhood cancer research. So this is a snapshot of our first release of the CCMA. And as you can see, we've got about over 400 lines received, of which over 200 have passed through our pipeline. We typically get our lines from across the world, and currently our efforts are really quite a collaborative effort involving 10 countries and over 38 collaborators. If we look at the types of cancers that are currently contained in our atlas, a majority of cell lines represent brain and CNS cancers, such as diffuse intrinsic pontine glioma, as well as medulloblastoma and ATRT. However, we have representation of other types of pediatric high-grade cancers, such as osteosarcomas and neuroblastomas, as well as some of the more rarer types, such as ETMRs. You can see year after year, we're receiving more and more cell lines locally and across the globe, and we hope that we're going to be the one-stop shop for childhood cancer research going forward. So as I mentioned, one of the mission statements of our program was essentially to generate cell line models from tumors which are poorly represented in the literature. We recently published our experience across the first 7 years in this STAR protocol paper, where we attempted to outline what we did and standardize a methodology, including the type of enzymes and the way we dissociate the material, as well as media conditions, because a lot of these cancers don't really have well-defined conditions in which they grow as. So across this time, we've identified some issues that we're still troubleshooting, and I think are issues that are faced around the world in this area. So the first issue we've seen is the fact that tumor type is a really important indicator of whether we're able to successfully establish a cell line. So we can see for some of the more high-grade malignant CNS cancers, such as DIPG, we are actually quite successful in establishing them. However, some of the rarer types of cancers where there isn't really an established protocol or media condition with which to establish a line, we really have quite variable results in establishing these types of cultures. A second issue that we've faced in establishing cell lines is the fact that a lot of cell lines seem to grow out in conjunction with non-malignant, what we've called fibroblast populations. So these are tumor-associated fibroblasts, we think, which lack characteristic alterations seen in the primary tumor. And so we can see this quite distinctly in our molecular profiling, which I'll show in the next slide. And then the third issue that we've identified is the fact that the way cell lines are established really dictates what genomic events are maintained in the culture. So this cancer discovery paper from Chris Jones last year showed that if they established a cell line in 2D versus 3D cultures, they had differing ability for that cell line to maintain a PIK3R1 mutation and therefore a differing response to the same therapy. And this is despite the fact that it came from the exact same patient. So together, these really highlighted the need to precisely annotate our cell lines and make sure that the models that we included within our CCMA really closely recapitulated the original tumor. So to make sure of this, we took a two-step approach. So on the left is an oncoplot of the pediatric gliomas included in our cohort. And the first thing we did is essentially compare the genomic profile of cell lines established in our lab to the genomic profile from their primary tissue. And so we could do this for certain cell lines which had characteristic mutations. So for example, for pediatric gliomas, the majority of them harbor mutations in their H3, in histone H3. So being able to match the cell line histone H3 mutation back to the corresponding tumor enabled a verification that this was an accurate representation of the primary tumor. However, as you can see, there's a subgroup of pediatric gliomas which actually lack H3 mutations and lack really characteristic canonical mutations to compare against. So in cases like this, we also took a secondary approach, which we based around multi-omic factor analysis. So this allows integration of multi-omics, in our case, DNA, methylation, and RNA, to cluster cell lines according to their identity. So shown here on the right is a new map of our MOFA analysis, and essentially we can see gain two things from this analysis. We can see that the different tumor types cluster quite closely with each other. So we can see ATRTs at the top cluster quite distinctly from gliomas at the bottom in green, but also we could cluster tumor-associated cells, which are those sort of fibroblast outgrowths in our culture, quite distinctly from tumor cells. So we believe the cell line models included in our CCMA are valid representations of their corresponding tumor types. So the two other aspects of our program was performing high throughput drug and CRISPR screens. So just briefly, our drug screens across our first 6 years of the program involved over 2,000 drugs across 3 commercially available libraries, the FDA library, the kinase library, and the Cambridge library. So we performed drug screens across 150 cell lines using a single dose, 1 micromolar. And despite only using a single dose for these screens, we were still able to identify a large proportion of drugs which had some selectivity. So you can see on the right a density profile of some of the classes of drugs that we had in our initial drug screens. And we saw around 1/3 of them showed selectivity as demonstrated by their biomodality in their density plots. The other aspect that we're interested in is annotating where we can biomarkers which predict drug response. And for example, if we look at single feature biomarkers which predict drug response in our CCMA, we could see some examples of this occurring. So anthracyclines, which is a class of chemotherapy, was highly predicted by p53 mutations, whereas dihydrofolate receptors was predicted by tumor type and were very effective in osteosarcomas compared to other pediatric cancers. However, the ability for these sort of single feature biomarkers to predict drug response really was in the minority, emphasizing that there are likely a multitude of mechanisms at play which dictate whether a drug works or not, and a more complex approach is needed towards defining biomarkers of response. Since performing our initial drug screens, we've now moved on to our 2nd generation of drug screen, which we've called the Australian Library of Pediatric Anti-Cancer Agents, or the ALPACA drug screen. So this drug screen was derived from our initial effort screening over 150 cell lines. And in our ALPACA compound library, we've got 732 drugs, which have known efficacy against pediatric cancer types. And some of the things that we're aiming to do with this more specific drug library is screen it against drugs currently in clinical trials to identify combination therapy opportunities, which seem to be more of a priority for clinical trials going forward. So just briefly, the other aspect of our program that we do is perform targeted CRISPR-Cas9 knockout screens. So these screens use a in-house built library comprising of just over 350 genes representing many known targetable oncogenes or drivers of malignancy. And the genes are performed across 21 to 28 days. So the graph on the right shows a representation of the results that we get from this sort of screening approach. And we can see that we identify many different factors which seem to be highly dependent. However, I'll let Claire go into that a bit more in her section. So I'll just finish with the next few slides showing some of the ways we're using the CCMA dataset for discovery research purposes. One of the foundations for precision medicine is using genomic features to inform therapeutic approach. And so shown here is a representation of p53 and all the different mutations which can occur along the gene. So for many of these sort of hotspot mutations, we actually have really good annotation of whether this is an indicator of a loss of function or gain of function, and whether or not this predicts drug response. However, mutations can occur across the whole gene, and oftentimes we really don't understand what the effect of these mutations are. So do these mutations indicate it's more specific, more sensitive to a drug, or do these mutations indicate that it's less sensitive to a drug? So we thought using our multidimensional characterization of our cell lines, we could integrate whole genome sequencing and CRISPR knockout screens to help prioritize these variants of unknown significance. The approach we took was quite straightforward, and it was based on the fact that we quickly realized that when a cell line had a pathogenic mutation, so this would be, for example, a gain-of-function mutation in a gene such as PIK3CA, when you knocked out that gene, we could see loss of representation in our CRISPR screen, so essentially cell death. But when a cell line had a benign mutation, we found that when we knocked out that corresponding gene which harbored that mutation, there was no corresponding loss of representation in our CRISPR screens. And by quantifying this, we generated essentially a score which we've called the likelihood score, which indicates the likelihood that a variant is actually gain-of-function pathogenic. So just to summarize, across the first 100 cell lines that we performed targeted CRISPR screens, we could identify 85 novel mutations that have previously been overlooked. So one of the examples that we were really interested in pediatric cancer was PDGFRA. So PDGFRA is one of the most commonly altered genes in childhood CNS cancers. However, mutations can occur across the whole gene, and in a lot of cases, these mutations really have quite poor annotation. So we found, I think, 10 mutations across PDGFRA across our cohort of pediatric gliomas in the CCMA. And by using the likelihood scoring method, we could annotate whether any of these were pathogenic or likely benign. So interesting, P250R was a mutation which hasn't been annotated previously as being pathogenic, however, scored very highly on our pathogenicity likelihood scoring system. So to validate whether or not this mutation actually was pathogenic, we took cell lines with either wild type PDGFRA, a known benign mutation in PDGFRA, a known oncogenic gain-of-function mutation Y288C in PDGFRA, or the novel mutation P250R, and tested whether this mutation drove ligand-independent signaling. So as you can see, in the wild type and benign mutation cases, in the absence of the ligand, there's a very minimal signaling through PDGFRA as indicated by the lack of phosphorylation, which is quickly regained after stimulation with its cognate ligand. In the case of the known oncogenic 288 variant, we could see that there was maintenance of signaling independent from the ligand. And in our P250R cell line, we saw the exact same thing. So this was interesting to us because this essentially allowed us to bin mutations into potentially benign or pathogenic, allowing more precise annotation and prediction of whether that mutation would actually predict drug response. So now I'll hand over to Claire to describe some of the other approaches we're taking.
Thank you, Paul, for the introduction to the program. I'm Claire. I'm the bioinformatician for the program. And this is a tech talk series for MPCCC. So I'll be focusing on the data set of CCMA and how we can use it to generate some hypotheses for our research and also some translational clinical implications. So firstly, I would like to talk about the highlights of our multi-omics programs compared to all other multi-omics pipelines. We have functional genomics, and we think integrating functional genomics in this multi-omics dimensions really help to fill in some major gaps. So on the level of different pediatric cancer types, we use the functional genomics to map out the landscapes of the enriched or unique genetic dependencies, which might result from their distinct molecular drivers underlying each cancer type. And with such a rich collection of our data set, as Paul suggested, we have collected a decent number of models. You can see for as a demonstration here, ATRT is an embryonal brain tumor, and we also have the largest collection of the high-grade gliomas and also osteosarcoma as some other solid tumor indications. So this really enables us to map out the enriched genetic dependencies in each cancer type. As you can see as example, in the high-grade glioma group, we have several genes that have matched targeted therapies. These are key kinase signaling pathways in the tumorigenic network, including MAPK1, FGFR1, PIK3, PIK3CA, and also HDAC2 is a histone regulator, and MCL1 in the apoptosis pathway. We also identified a unique metabolomic enriched pathway or function in the ATRT group, and we found this SDH A, B, C, D genes, key regulators in the OX-FOS pathway, the energy regulation functions. And YES1, which is a SRC kinase family, is an interesting target in the osteo samples. So these novel targets or biomarker can support some hypothesis for each cancer, major cancer types. So secondly, how we use this functional genomics is, I think is more interesting here is to look into the different molecular subtypes defined in these major groups. We have decent number in each subgroups too, so this really enable us to do some meaningful comparisons between them. So we have mapped out our samples to group 1 and group 2 ATRT, which have been characterized from previous studies. Distinct dependencies in the developmental and epigenetic regulatory genes have been observed in the group 1 models, which have been characterized happen in a younger age and also different locations in the brain. And worth to mention, KMT2A, this gene have targeted therapies in trial for the leukemia patients. So this is really a great potential for repurposing this drug for this type or subtype of this tumor. Similarly, high-grade glioma have been officially subgrouped by the WHO classification system based on the histone mutations. So we have H3K27M mutated midline glioma and H3G34 mutated hemispheric glioma, and also other H3 wild type high-grade gliomas. So we have identified epigenetic enriched dependencies showing in this red, highlighted in this red sections here. In the H3K27M subgroups. Further, if we look into the two different isoforms of K27M, we also found distinct genetic dependencies or vulnerabilities. H3.1 showed more dependency in the BCL2L1, which again is the apoptosis pathway, and H3.3 K27M models has distinct dependency in HDAC2. So we also have several histone inhibitors. So this will be a good evidence or points to perform the secondary validation. So after the subtype level, the third level, we can come down to the individuals. So this is how we can use functional genomics to contribute to precision medicine. So from the molecular subtypes, we can identify the major types and the subtypes of each patient. And at present, precision medicine initiatives mainly rely on DNA sequence to identify the DNA mutations that drive the tumor genesis. So this serve as the gold standard to forecast how patient will respond to targeted therapies, and then we will, based on this information, to recommend targeted therapies or trials for this patient. However, it's unfortunate, we all know children's cancer are with very low mutational burdens, unlike the adult counterparts. So the challenge here is really using DNA mutation as a biomarker of response to these targeted therapies is really poor. So this highlights the need. We need to go beyond this DNA mutations to identify potential biomarkers that help us to find the right therapies. So in this plot here, really demonstrate the current pipeline. Once we identify a variant, the curators will mainly rely on the database or knowledge base, annotating all these known pathogenicities that mentioned by Paul before too, to make recommendations to the clinicians or oncologists. So what we want to do, if we during this curation process, if we do the functional annotations to these mutations of these genes, this will enhance the recommendation of the targeted therapies. So there are 2 cases so far from the local patients that enrolled in our program. The first case is demonstrated by Paul already. So this is a 7-year-old patient with DMG enrolled in the Sydney Children's Hospital. And the patient's tumor had a novel PDGFRA mutation, so P250R. Using our method, we have approved this novel mutation, which hasn't been able to report to the clinicians, actually have oncogenicity. So adding this annotation from the functional genomics really enhanced the recommendation of PDGFR inhibitor to this patient. And indeed, I think the trial recommendation for this patient at the end is the PDGFR inhibitor, aligning with the amplified PDGFR expression and also this mutation. Similarly, another DHG patient case enrolled in the Royal Children's Hospital has this TMPRSS2. Again, this is a variant of unknown significance. So this gene encodes a transmembrane protease, and it is actually a hallmark gene with another fused protein in the prostate cancer. And also this protein has been widely studied in the COVID-19 disease, I think. So there are some drugs that can be repurposed for this gene. And we found that by having this mutation with 319M mutation, really make this sample more dependent in this gene. So again, we are validating using the same method here as demonstrating the PDGFRA case. We try to see whether this mutation is directly causing the stronger genetic vulnerabilities. So I think the take-home message from the major cancer type, the subtype, and the individual cases really demonstrate adding the functional genomics in the multi-omics pipeline help us to better link the genomics and the response to targeted therapies. So the next part, I would like to talk about some proof of concept study. We're using AI or machine learning, deep learning technology to make some predictive models for the genetic dependency or drug responses. So in order to effectively integrate machine learning into our research, it is crucial to have a systematic understanding of the data. One approach we use is the MOFA, as mentioned by Paul, which really allows us to summarize tens of thousands of these molecular features into just 10 dimensions. So with this tool, we can measure the contribution of each data point to the variance observed in these 10 latent factors. Through the application of MOFA, we made an quite exciting discovery or approved our lines really recapitulate the tumor they derived from. So this really affirmed the reliability and consistency of our data set to do more research into it. For our research, we wanted to build predictive models, as I mentioned, for gene dependencies and also drug responses. But a more important question is not only the predictive model itself, we also want to rely on this method to help us prioritize or extract the inner working of these molecular features from RNA, from DNA, from methylation, and the clinical informations. So the random forest here serves as a valuable tool to establish these connections, help us to understand these networks. By visualizing and interpreting these models, we are interested in the factors that contribute to this model. So by diving into this black box here of machine learning, we can then to find the potential biomarker. And again, very importantly, is to validate back in the lab that is the true biomarker predicting the gene dependency and drug response. So as an example here, applying this method to our drug data really help us to navigate to some biomarkers of interest. We have built confident predictive models for 53 drugs from our first generation of drug screen, and these drugs are showing some selectivities between some samples. So in the models for these 3 drugs, they are all DHFR inhibitors. We found a common feature as LC5A6, which is a regulator in the biotin transmembrane transporter family. So this really in the top range of the contributors for this prediction. And then because these drugs are like gold standard, or I mean standard therapy in treating osteosarcoma, we also found these genes are highly expressed in the osteosarcoma models, showing these pink dots. And the secondary validation back in the lab can distinguish the high expressed versus the low expressed. And then they have different response to this DHFR inhibitor pralatrexate. So this is an example, and using these methods to our functional genomics, children's cancer, we want to use this to build a children's specific predictive model, because children's cancer are not mini version of adult cancer, and the comparison of dependencies between the adult high grade and the pediatric high grades, for example, showing on the right and mentioned by Paul before, really reveal that pediatric, even for the same cancer indication, pediatric patients have specific dependencies, so genetic vulnerabilities in children. And then more importantly, except for these distinct genetic vulnerabilities, even if there's a common dependencies between the children and the adult cancers, we want to ask or investigate is there any difference in the underlying tumorigenic network that support these genetic vulnerabilities? So using the machine learning really help us to dive into it. So by building these models using our data set, because it's a pediatric specific data set, and also compared to the adult-focused DepMap data set developed by the Broad Institute, we have discovered that potential markers for a given gene dependency can differ significantly. So all the orange gene on the top panel are the examples where they share the same underlying network. So MDM2, for example, is pretty much determined in by the TP53 status in both the adult and pediatric situations. However, the purple ones are the ones, even they are commonly predicted, however, the underlying biomarkers differs significantly. MCL1 dependency, as an example here on the right, really shows that in the adult on the top, BCL2L1 expression really help us to determine whether this model will depend on MCL1 and then use MSL1 inhibitors. However, in the pediatric, we don't have very strong single biomarker that can determine MCL1 dependencies. So further, we found that pediatric high grade showed enriched dependency compared in PIK3CA, this gene, compared to the adult high-grade gliomas. So using the same method, we all know that PIK3CA mutation really is the biomarker for the dependency or the vulnerability of this gene and also this pathway. And we pick PIK3CA mutation as the leading contributor in this prediction. However, in the pediatric scenario, showing in the bottom here, using our data set, is different. And we actually validated using our drug screen that the lines we selected here showed pan response to PIK3CA inhibitors. And if we look into more detail, we found that a third of our pediatric cases can't be, the dependencies to PIK3CA can't be explained by any genetic mutations. The others, 20% of them are directly linked to PIK3CA mutation, and half of them are linked to a more generic mutation in the pathway. So this really reinforced the need to use pediatric data to model cancer vulnerabilities as the biomarkers. So we can't really use a lot of the data set are available for the adult cancer cell lines or adult cancer tumors to make AI predictive models for pediatric cancers. So moving this forward, I've been working with Samisa and David Dahl from the IT faculty. So we're trying to develop some deep learning models in this. So conventional machine learning like random forest is relatively more supervised method. We need to do some dimension reduction, as I mentioned. However, deep learning has more power in terms of making accurate predictions. So he has been using deep neural network and also autoencoder to architecture these models. So there's some preliminary data from the deep learning models. Some key message here, by using multi-omics profiles, he has been making the strongest prediction, and the correlation has achieved, average correlation has achieved 0.8. So this is really exciting results. And another key message from this is if we're only relying on DNA mutations, for example, point mutations and point mutation plus copy number variations, we couldn't really get strong predictions of the drug screen or gene dependencies. So this again highlights DNA mutation itself is a poor biomarker of the drug response or genetic dependencies. And comparing the deep neural network versus our published random forest models, here these are the genes as positive controls. So both models have been able to make strong predictions for this gene. And you can see on the top are the ones that have been outperformed in the deep neural network models. So by diving into these models, we are facing some challenges in here. Firstly, we're trying to integrate or harmonize our cell line data with the tumor data itself. So at the end of the day, it's the tumor growing the dish. It's not mimicking, having the microenvironment, all these 3D structures. So we're trying to use some data or statistical way to integrating our model with the tumor data. And secondly, we want to, again, like random forest, we want to open the black box here. But during this process, we found there's no, this outperformed genes that I just mentioned in the previous slides. There's no single biomarker or even like handful of biomarkers as like sufficient to make predictions. So maybe at the end of the day, we have to rely on a whole profile to make strong, to capture the picture, the molecular profile as a whole to make strong predictions for drug and gene dependency predictions. So at last, I would like to just do some demonstration, very short demonstration for our CCMA database and also the interfaces that we have built to share our data set with the field and the research community. So this is the dashboard where we can track our progress. So this number is a bit more than the one Paul showed. We have nearly 500 lines, and yeah, we are really busy in the lab to get all the lines through the pipeline so we can generate more data and support more research here. And also Vikesh has helped us develop this portal to really share the data behind the data set, not only where we, how much we have achieved. So this is an interactive portal where you can search your gene of interest. And then from just simple clicks, you can find all the RNA, DNA, CRISPR drug data associated with this gene of your interest. And from here, you can generate a lot of hypotheses and design your experiments. So future directions of us, I think, of course, we will do more lines and we will have more data modality. So we are now doing whole genome CRISPR and alpaca drug screen, as Paul mentioned. We are having phospho and total proteomics and immunopeptidomics in the multi-omics dimensions. And also we are collaborating with Zero and UCSF clinical teams to contribute to this curation pipeline, as I mentioned, and also more prediction AI models as exciting research discoveries here. Yeah. Thank you, everyone.
Thank you, Paul, and thank you, Claire. This is so much work. It's really incredible. Well done. Do we have any questions for Paul or Claire or both? You can just unmute and ask your question if you like. Have a comment. Really great work. Thanks so much. While we wait for questions, maybe I could ask one. So in your study, you've generated the whole genome sequencing data, the RNA-seq yourself, and the methylation as well. So you've been able to control for the type of for how the data was generated, and everything was pretty it was done the same way, right? And I was wondering if you had any, it's actually a question. It was done.
Yeah, so is your question like this pipeline for all these samples are consistent? Yeah, we're trying to control this, but it's been 7 years. I think there are two, for example, RNA, there have been two major upgrade because HiSeq now is stopped everywhere. We have to now using NovaSeq platforms to sequence the read.
Right, yeah.
And something like we upgrade to stranded RNA now. I think for the first 2 years we used unstranded. And for example, whole genome sequencing pipeline, now we are trying to bring it up to HD38. So this really, I try to make it consistent. So this also involves we need to reanalyze a lot of data and try to do batch effect correction things. But yeah, it's not completely avoidable, even though we're trying to control the technical variance. But yeah.
Yes. So my question was, what sort of advice would you have for people who are trying to harmonize data when it comes from different sources, and what was the main challenge that you encountered when you for instance, when you went back to reanalyze or incorporate a new type of data?
Yeah, that's a very good question. So remember in the MOFA analysis, before you do you need to look into the data, like in all these latent factors, is there anything correlated with the confounding, technical confounders, and do include these confounders in your analysis. So, which I mean, you provide the modeling, for example, the AI modeling between these samples, what's the RNA difference, what's the whole genome, the different batch effects. So in this way, we can try to not prioritize the biomarkers that correlates with these batch effects. It doesn't mean they are not true. It just because we can't control it completely, right? So yeah, we try to, on an individual modality level, we use some statistical models like RUVs here and there to correct the batch effects and do some normalization as much as we can do. And another way, for example, if it's completely different data sets, we want to harmonize, for example, between the tumor data, between us and DepMap, we are investigating more powerful, more robust statistical models too. So I talked to some statisticians, they mentioned several very complicated stuff I haven't been able to understand, but this is a known issue, like because all this large data is trained in everywhere. So I think it's already been a hot topic in this area too. Like at the end of the day, you can't avoid them. You just need to, how can we avoid this confounders misleading the decisions or risk analysis?
Yeah, I might talk to you after this.
Yeah, thank you.
And see what your advice is on this question. So we have a question from Legana regarding your AI model. It had 500 features, which seems to be quite a lot. Just wondering how large your data set is, how many rows, and how are you dealing with model overfitting?
For the deep learning stuff, so 500 features is not actually many comparing to what from methylation we have like 800,000 probes. RNA, we have 20,000 genes. Whole genome, we have more than that. So I think the first step in the random forest, we use Pearson correlation to prioritize the input to 1,000 each. And in the deep neural network, Samisa used a key, I think his method is key best or best key selection, which is a statistical model to prioritize things first and then fit into his model. Random forest doesn't have the overfitting problem, but I'm not too sure about I think to answer this question, because for the deep neural network, the validation model also holds quite strong accuracy predictions. So I think we are really controlling the overfitting things in this way. We subdivide our cohort into a good number of validation and learning testing set. So yeah, does it answer the question? Thank you.
Thank you. Do we have any other questions? In the chat, or you can just ask a question directly. I think that's it. Well, again, thank you very much to both our presenters for coming today and talking about this wonderful study. I'd like to thank everyone for attending, and we'll see you again next year for more MPCCC Tech Talks. Thank you very much. Bye.
Okay. Bye.